So from a little bit from the biology perspective of how do we go about selecting the probes, what are the experiments we need to do, like in vitro, like in vivo, how do we go about testing a probe, or how do we use an already well-established probe. So as an outline, again, T1 is important. That's going to be, I think, a recurring theme throughout the course. Applications, so far, how hyperpolarized C13MR has been used. Metabolisms, what are the requirements of a successful probe? Probe testing and utilization in the different systems, just ex vivo considerations. I'm going to go into a little bit detail on the bioreactor, which is a system that we use here at UCSF, both in the Ronan group and in our group, to do cell studies and in vivo experiments. How do we set that up? And then a little bit into the choice of probes, in the sense the different positions that can be labeled, because of which we can study different pathologies, or different organs of interest have a very specific metabolic pathway that's of interest. And then a very, very brief overview of some successful probes and specific applications. It's more or less like a very quick review, and even though the field is like relatively new, it's like exploding, and there are already more than 25 reviews. There's pretty much at least 2 reviews that come out every year. And so I've tried and listed some of the references as I go along, as I give examples for the different applications and things. And I can put up a more comprehensive list of reviews that's out there on the website. So T1, why is T1 important? Because the signal, the hyperpolarized signal, needs to last enough so that we can actually image it. So from the time of dissolution to the time of injection into our system, be it cells or animals, up to the point of metabolism and to study its products. And so by the time 3 T1s are over, you have only 5% of the signal left. So again, remember what Peter was saying, everything you have to do is like fast, 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 really fast. You use up to maybe 10 or 15 seconds after dissolution, you inject it into the animal, and then you wait a certain period of time to maximize your signal from your substrate into your products, again, depending upon the organ, depending upon the probe that you're studying, and then you acquire data, either a single time point or a dynamic measure, more of which Jeremy will cover in the pulse sequences and acquisition strategies. So what I have here is simulated pyruvate and so we have the pyruvate T1, which is about 46 to 48 seconds at the 11.70, and then the lactate T1, which is shown in the red curve, which is about 30 seconds. And so the T1 is extremely field dependent. So this paper here shows the change in the field as you go from the polarizer to the magnet. Different groups have different setups, and so the Switzerland group has a dedicated chute, so to speak, where it delivers the hyperpolarized substrate directly into the magnet. And so sometimes what we do here is use a holding field or use a magnet to carry a substance over up closer to the magnet in case the substrate has a really, really short T1, like glutamine, which has a T1 of about 23 seconds at 3T. Again, all the T1s I'm listing is in the magnet, so it's fixed for that field. And we can do extensive studies if we need to map out the T1s in these low fields. But the T1s listed are very specific, and all of the T1s that I've listed here are in the solution state T1s, right? In vivo, obviously, it's going to be much shorter due to binding, due to paramagnetic relaxation in the blood, and a host of other interactions. And also, there is not a very set specific relationship between the field and the T1. So the general understanding is that for the carboxylates, which is mostly the carbons that are labeled for the hyperpolarized studies, the T1 increases as we go down in the field. But for probes like the keto sugars, like the fructose and the glucose, the T1s increase with increasing field. So I don't understand the exact physics behind the dependence of the field on the T1, but I think it's in people are trying to understand that. And so this hyperpolarized has been used for receptor ligand binding. The reason I list that first is this is actually the most uncommon use of this. There's only been 2, and although not in vivo or biological applications, PET is obviously the winner when we're trying to study the receptor ligand binding application. So this is shows a T2 weighted image of different vials with different concentrations of cyclodextrin. And when you add benzoic acid, so this is hyperpolarized benzoic acid, you can see the loss of signal with increasing cyclodextrin concentration. Right over here is 10. And so when the hyperpolarized benzoic acid binds to cyclodextrin, because of the large molecule, the correlation time is now larger, and so that's the reason why you lose signal. And that's why you have no signal with increased binding with higher presence of cyclodextrin. Although this application has never been tried with hyperpolarized MR in vivo. And the most obvious utility is in metabolism. Pyruvate is the single most commonly used probe. It's like the FDG of PET. And of course, perfusion and permeability. Cornelius has done, and Galen has done quite a lot of work comparing the different substrates, such as HMCP, which is also known as the HbO1, the butanol, and urea. So these 3 compounds, as you can see, the different images vary in their perfusion or permeability of the different organs here, through the tumor and through the kidneys, and I think through the liver. And so I've listed the references in full over here, and I can also list them separately as I go through. So what makes a probe successful when you want to study metabolism? So this is a more recent review in the last month, I think it came out, by Miriam, and it's very neatly charted out, step by step. How do you go about developing a probe? So I think this is very useful if you want to think about a new mechanism or a new pathway that you want to interrogate, and where do you start? So the very important thing for metabolism is extremely rapid enzymatic reaction, right? So conversion. And that's not just the only thing that you need. You also need a quick transport into your cells or into the mitochondria, or your location of where the metabolic pathway of interest is. So a lot of cases, even if you have a rapid enzymatic reaction, one of the biggest hurdles is for the probe to get to your region or location of interest. For example, here I've shown glutamine. Even though glutamine has been shown very successful, not very successful. In some cells, it's worked. There has been like one study, even though this probe was first published in 2008, in one single in vivo experiment with barely any mention or image shown. And the reason for that is the glutamine transport into the cell is a major limiting factor. So this figure here from Gallagher et al. Shows that in the second. So the first bar graph is the glutamine in lysed cells. And the darkened out bar graphs is the glutamate produced. So this doesn't jump out at you, but with the lysed cells and with the intact cells, if you look at the glutamate to glutamine ratio, it's actually twice as much. And that right there tells you you're losing 50% of your metabolic signal that you would otherwise have if not for the transport. So again, this is important when you're trying to test your probe that you're making from scratch or some new system is you want to test it in the system that it's most relevant to, including all these different components. I elaborate further on what I mean by that. And the other important thing is preserving the polarization as it goes through maybe 1, 2, or n number of enzymatic steps or reactions. So this is, again, another example to illustrate, similar to the benzoic acid experiment. Here you have a salicylic acid. And the ascorbate is just shown as a reference here. As you can see, the green labels 1 and 3 arising from the salicylic acid have specific resonances. But when you have a serum albumin present with this hyperpolarized salicylic acid, the signal from the salicylic acid goes away. Again, it's the rotational correlation time that changes and wipes out the T1 or your hyperpolarized signal. So it's important that as your substrate goes through all the different enzymatic reactions, the polarization needs to be preserved for you to measure everything. And another interesting factor to keep in mind is the metabolite pool sizes. This specific example is for pyruvate. So it's understood the most, and the reason most works show that there is no net production of lactate. The lactate we are observing is mostly a function of chemical exchange with the unlabeled lactate pools. So if the product endogenous pool size in your system is low, the signal readout is going to be low. For example, if we were studying conversion of pyruvate to lactate in a resting muscle, where the lactate levels are going to be very low, you would get no signal whatsoever, right? So that's because the lactate pool size is small and the exchange is not going to be feasible. Again, the basis behind it is that there is no net lactate production in these systems that we have predominantly been studying. And so that's what this graph shows. So the blackened out plots are the experiments done with pyruvate 80 millimolar injected into the animal with 80 millimolar of C12 labeled lactate. So what you're doing is you're increasing the pool size. And lactate also gets into the site of interest in the exact same way as pyruvate would, through the MCT1 transporter. And so that's, again, very specific to this particular substrate. So as you can see, in 3 different regions of interest or pixels of interest for the liver, the vascular region, and in the kidney, this was done in a rat. They showed that in the kidney, there's as much as doubling of your lactate signal by giving a cold lactate infusion along with your pyruvate. And so liver, there is only about 20% increase in your lactate production. But so again, it depends on which organ you are interested in. So these are all the different factors that you want to keep in mind when you're trying to test or develop a new probe. Yeah. It's been tested and shown proven for lactate. This again is... In this case, the pyruvate and the lactate use the same transporter. So yes, that's again... different transporters, then adding an excess of unlabeled product may not... Not during the shot maybe, but if you give it, pre-dose it, and you give it sufficient time to get to that region, which doesn't have to go through the same transporter. And so our star probe, the C1 pyruvate, has all of these characters, right? So it has long T1, it swiftly crosses the membranes, there's rapid enzymatic conversion, and there is preservation of the polarization. As you can see, we've seen even up to glutamate going through the PDH enzyme. And this has been applied to study cancer biology, treatment of cancers, cardiovascular pathologies, diabetes. It's been used to study different organs, liver, kidneys, the brain. And so once you've developed it, how do you go about testing it, right? So the very basic, the single most simplest way of doing things is things in a tube, right? your enzyme systems in a tube. Or if the example shown here is that of the pH probe bicarbonate, which is used to measure interstitial pH. So these are buffers at different pHs with and without carbonic anhydrase, which is pretty much the fastest known enzyme that's used to convert bicarbonate, interchange between bicarbonate and carbon dioxide. And this was the very first study that can very simply tell you whether or not your probe is working. This probe is intended to show pH, and with this experimental setup and correlating it to a pH electrode test, you can tell whether or not your probe worked, right? So you inject hyperpolarized bicarbonate into these tubes with differing buffers at specific pHs with and without carbonic anhydrase. The other way of studying these probes is if it's a specific enzymatic pathway, then you have different enzymes in a tube. Or you use cell lysates. If the enzymes are not readily available, what you would do is take a cell and lyse it and put it in a tube and do the experiment. But one thing to keep in mind when you do these test tube type experiments is, again, you're not taking into factor all the disadvantages of the transport of the probe into the cell or even into the mitochondria, depending upon where your enzyme is located. And the other thing which is not always obvious is all the different cofactors that are required for your enzymatic step to happen. Especially when you do cell lysates, you have to prepare your cell lysate extremely carefully because degradation of all the different constituents happen extremely rapidly. So you can have protease inhibitors in there to prevent the degradation of all the cofactors. But even with cell lysates, when we try and use the LDH, we almost always have to add more NADH in order for the reaction to occur. Because you would, again, have very, very low signal otherwise. So we tend to do most of our testing here with intact cells. So one of the, again, a common way of doing things is a cell slurry or just trypsinized cells in a tube, which is, again, not physiologically maintained. So in order to prevent the cells from lysing or dying, we keep the cells in a tube really cold until the time of the experiment. And then we need to have, even for a 5 millimeter NMR tube, we need to have at least 30 to 40 million cells. And that gives us exactly one experiment. And we all know hyperpolarized experiment takes at least 10 tries before maybe it'll work once. So that's not a very efficient way of doing things. And so what we do is cells in a bioreactor. We are fortunate enough to have really nice systems here. Both the Ronan group and our group uses that routinely. And so I'm going to go a little bit into more details about the bioreactor. So a bioreactor is a system that keeps the cells physiologically relevant condition. So it's continuously perfused with media or nutrients. It's maintained at 37 degrees as if the cells were sitting happily in the incubator. And it's supplied with oxygen. So there's efficient oxygen delivery to the bioreactor. So some unique features of one specific system I've worked a lot with is it's precision engineered. We build it from scratch over here, and it's susceptibly matched. So we get amazing homogeneity, and we can resolve peaks that are 3 hertz apart. So that's the amount of resolution that we get. And it minimizes the amount of sample that's required, and we can reuse it over and over and over again. And we've maintained cells in the bioreactor, observing it in the magnet for 72 hours. And one way to assess the viability, since we are able to keep it in the magnet, is using phosphorus. So if we use phosphorus in NMR, you use beta NTP peaks, which is a measure of the ATP content of your cell, which is the first thing to go, by the way, as your cells die. And so we measure ATP, and that can actually be related back to the specific number of cells in your active region of the coil or in your experiment. And we can do multiple experiments. We can go between experiments. So we usually first start off doing a phosphorus experiment to make sure that the cells are alive. Then we do a C13 experiment, switch back to phosphorus, and we can keep doing this all day long. We've done as many as 10 shots in a single bioreactor, and the cells do just fine. But one word of caution is the way you prepare cells can be very different depending upon the type of cell that you want to use. I'll go a little bit into that in the next slide. And so just to show the outline here, so you have a circulating loop of media that's flowing into the cell, and then you have, in our bioreactor, we have a plug at the bottom to minimize the volume of cells that you need. So you can get away with as low as 8 million cells is what we've done. And then you have a water bath that your media flow is completely enveloped in. So it always keeps the media at 37. And then you have an extremely efficient gas exchanger which supplies the oxygen to the cells. So this could be considered actually quite a laborious process of preparing the cells for your experiment. So the way we do our cell preparation is encapsulate them in alginate microspheres. Ginny actually prepared a video of us doing the entire thing, so we'll put it up on the course website soon. So there are different components to it. It's actually nothing new. It's been around for 15, 20 years, this electrostatic encapsulation. So what we do is once you lyse your cells, what's shown here is just alginate with dye in it so you can visualize it. So we trypsinize the cells, put it in alginate, and it's a really viscous stuff. So you draw it up in a syringe and you let it drop into this bath of calcium chloride. And the electrostatic difference between the electrodes, it's hooked up to a really high voltage source. And you keep the needle and the water bath you apply a difference between 3 to 4 kilovolts. And then you let it go drop by drop into your calcium chloride bath, and you can make the beads. And so the potential difference between the two and the rate of the power injector decides the size of your beads. We try to keep the size of our beads anywhere from 300 to 400 microns. One thing is to prevent it from flowing out of the tube. We have a stopper, and that has holes to let the media flow, but we don't want the beads to occlude those holes. So we want to keep it in a specific size. But a more important factor when you make these alginate beads is the diffusion limit. So in vivo, 150 microns is considered the diffusing limit before the metabolism starts kicking in, that oxygen is not delivered beyond that region. And so we try and keep our bead size no more than 400 microns, so to enable efficient nutrient diffusion into the alginate beads, which has the cells in it. Any questions? So this example here shows actually we were able to encapsulate just the LDH enzyme. So we bought LDH enzyme off the shelf, encapsulated it, and the dynamic traces actually shows you the pyruvate, lactate, and urea. We just had it in there so we could actually do some kinetic modeling of the input function and a whole bunch of other modeling parameters that can come off the urea. Because we do all of our bioreactors under stop flow condition, but the Runa lab always stop their flow. This is extremely important because, one, modeling is going to be very different. The interpretation of the signals would be then dependent upon how quickly or how late your pyruvate reached there, your input functions, and all those things. But recently, we've realized we have actually yet another big problem. But before I go there, one thing I wanted to mention is this is not the only way you want to prepare the cells. There is another way that is more suitable for certain kinds of cells. which is the microcarrier beads. So you have polystyrene beads, and the cells are grown on top of those beads. The reason why those systems might be more suitable for your cell is because the cells do not thrive in this 3D environment, because most of the immortalized cell lines are used to an adherent 2D type of growth. But once we put it in the 3D microsphere of alginate, it might be different metabolically even, not just physiologically. So depending upon how well your cells survive in this alginate beads, it may not be suitable for you. So most of the cancer cell lines, the renal and prostate cancer cell lines that we work with actually thrive very well. In fact, they actually thrive in the 3D culture. But some of the other cell lines we worked with, for example, macrophages, do not survive at all in this alginate encapsulation. So for those cell lines, you need to use the microcarrier beads. So before you even start doing hyperpolarized experiments, you need to evaluate whether or not these different modes, are applicable to your system. And so as I was saying, flow. Flow is important because in our specific problem that we encountered was the pyruvate that's converted to lactate actually exits the cell and it flows out, and we actually lose our product. And it exits through this transporter called MCT4, which is a biomarker of cancer aggressiveness. So once again, you want to think about the different experimental conditions for a known system about how you want to do it. So the way we were doing some of our experiments for renal cancer and prostate, we found that with higher the grade, more lactate is produced, but that lactate was flowing away. So the net lactate to pyruvate ratio was actually going down in the more aggressive cancers. It's just something that we ran into that might be helpful for you to keep in mind when you're designing your experiments. And so what we've shown is the in vitro or cell experiments. We can extend this to a little bit more relevant biological models, such as the patient-derived tissue slices that we've set up here in collaboration with Dr. Peel from Stanford. We worked with prostate and renal tissue slices, and we've shown that with a hyperpolarized pyruvate, the lactate produced increases with the aggressiveness of the tumor. And the example shown here is one using C1, C2 labeled pyruvate, and you can see the production of glutamate is actually more than that of lactate, even though it's 5 enzymatic steps removed from the lactate. So it just again goes to show the different pathways that can be studied with a single molecule if you design your experiment correctly. The other ex vivo type experiments that's very popular is perfused organs. I'm no expert in that, but those are like 100 times more difficult than the cell experiments. First of all, to surgically remove the heart or the liver and have it functioning or beating outside of the body and keep it, again, physiologically relevant to what we're doing, depending upon the things that you have to perfuse the organ with and then make it relevant to your study is not trivial. So moving on to in vivo considerations, some probes are just not feasible to make it work or test in cells. For example, the DHA probe, which is a marker of the redox capacity of the cells. We've never gotten it to work in cells. And the two publications, one from our group and one from Brindle, have just shown the in vivo data. That's because of the difficulties that's involved with the transport of DHA into the cell again and the T1s. And so you may directly just want to use an animal model to test your probes. Depends. So if you're doing that, then you really need to consider the toxicity of the probe, the solvent, the radical that goes into the probe, and all the different constituents that make up your probe. pH of the injecting solution. You cannot just put in 80 millimolar of pyruvic acid into an animal without neutralizing it. The volume of the injectables. Again, in vivo experiments can be time consuming. So you want the animal would be in the magnet for like 3 or 4 hours, depending upon the number of shots. And so you cannot keep injecting 300 or 400 microliters, considering the catheter volume and everything. So because one essential part of doing an in vivo experiment is putting a catheter into the animal. The most common catheter that we use is one from the tail vein. The other one, you can use the jugular, which is not trivial again, and can be really hard to keep it patent over time. And so with the catheterization, you need to keep the line patent for the 3 or 4 hours that you're doing the experiment. So you're continuously injecting saline to keep the animal hydrated, keep the line flush. And so all of that loads the animal with the fluid, and it's under anesthesia. So those are some physiological parameters that you want to really pay attention to. Then another interesting work was from the Stanford group was based on the depth of anesthesia. Just like any other functional MR concerns that you would have upon how that would affect your metabolism, this work showed that in the rat, the high and the low was the high and low dose of anesthesia. So that that determines how asleep your animal is, right? So our easiest readout is the isoflurane level and the respiration of your animal. And so what they did was they alternated the high and low dose of the anesthesia. And the different plots shows the different lines are from different animals. And you can see that the pyruvate actually changed with the different doses of anesthesia, whereas the lactate and the bicarbonate remained relatively flat. And this was all done in the brain. And what's important is, okay, so the lactate and the bicarbonate didn't really change, so we are home free. But that's not necessarily true, because in 99% of the way we present data is lactate to pyruvate ratio. Because we cannot get absolute concentrations as we otherwise would get when we do high-resolution NMR with metabolite estimations, with hyperpolarized data, we cannot get absolute concentrations. So in order to account for the differences in polarization, the differences in the time it took us to do the experiment, we always take the ratio, express our product as a ratio of our substrate. So lactate to pyruvate or bicarbonate to pyruvate is how we represent our data. So if the pyruvate levels are changing with the depth of anesthesia, then it's a major concern about how you represent your data or how deep your anesthesia is, and again, the organ you're studying. Because the same work showed that in the rat, the pyruvate levels didn't really fluctuate in the kidney or the liver. It was only in the brain that it changed.
Sorry, quick question. Yeah. So then, is this because of the change in the distribution? I mean, or like how the probe's getting to the target site?
That was not clear, but I think it has to do with the vasodilation, because under anesthesia, that changes. So the pressure, so the delivery is different. Mm-hmm. I think. I don't know, unless someone else has any ideas on why that would be the case. And so once we have all the physiological parameters set, what's important is, again, the animal models, the choice of study is. So I'm going to here highlight some of the issues that we've had to deal with in terms of cancer biology. So we are trying to work with more realistic models of cancer that are more popular out there, but not necessarily for imaging. So we tried to study tissues derived from patients that we've used in the bioreactor, implant that in the animals. That's a very popular model system to study vascular agents or vascular therapeutics. But that's really not suitable for imaging, because the tumor doesn't grow to a sizable range that can be imaged, especially for carbon imaging, which again, Jeremy is going to explain all the limitations, constraints that we have to work under, is the size. We need to have at least 1 cc of tumor in order to reliably get information specifically from the tumor, which we never were able to obtain with patient-derived tissue slices, unless it was extremely aggressive and it would grow within, say, 6 or 7 months. Even that took 6 to 7 months to grow. There have been some cell lines which grow actually extremely well. So it's obviously perfused. It doesn't get necrotic in the core. But during the time frame of a hyperpolarized experiment, the substrate do not get to the tumor because of extremely high interstitial pressure. So that specific tumor model would work great for a DCE type experiment, but it just will not work for hyperpolarized experiments. Again, mistakes from our experiences. We've tried several renal cell lines that have beautiful tumors that we've used with DCE, we've used with ultrasound imaging, but we could never deliver the probe within the frame of time that's required for a hyperpolarized experiment. We know that the pyruvate signal is there in all the other vasculature, but it doesn't get to the tumor. But the tumor was not necrotic, and the tumor grows beautifully within a given set of time into a quite large number. Another concern would be the location of your organs. So different tumor models could be orthotopic, could be just a xenograft. So the region where it is dictates also how you're going to acquire your data, the hardware available to do so. So some groups exclusively work with only subcutaneous tumors, because they use only a surface coil for their readout. They don't have fancy pulse sequences to localize their signal, which we fortunately have. And so the different models we've studied is not just our group, but all the different groups here is genetically modified models, xenograft models, orthotopic models of renal cells in the kidney, or brain tumor cells implanted into the brains. And so far, those worked very well. Again, when you're exploring with a new cell line, you need to first characterize the tumor growth and have a few animals before you can actually try a hyperpolarized experiment with it. So some of the probe considerations in terms of where and how you want to label, which specific label do you want to use for your study, depends again on T1, a recurring theme. The reason why I mentioned T1 is if lactate is, in your belief, that the best readout for the disease or therapeutic that you're studying, then you would always want to use C1 labeled pyruvate, because the C1 labeled lactate has a T1 of about 50 seconds at 3T. If you were to use the C2 labeled lactate alone and use the lactate as a readout, you're going to be really surprised, because it's as short as 10 seconds in vivo for the C2 lactate. So you have very limited amount of time to actually measure your probe. And so you're going to be extremely SNR challenged if you want to do lactate as a biomarker if you're using only the C2 pyruvate. But then different organs have different metabolic pathways upregulated. The pyruvate carboxylase flux is actually very, very low in most cancer cells. However, it's extremely high in the liver and in the fed or postprandial state. It's a very clear indicator of the upregulation of the pyruvate carboxylate, the function of that, as is shown in this work here by Merit et al. With just using the C1 pyruvate, you can observe the PC flux going to both the malate and the aspartate. But if you wanted to study the PDH flux, C1 pyruvate will not be a good choice of probe, because you're going to lose the C1 label as bicarbonate, and you will not be able to follow the TCA intermediates. And so in that specific case, you would end up using either a 2 labeled pyruvate or a 1,2 labeled pyruvate or a C1,C2 copol pyruvate. So that brings us to using copols of substrates. In the previous case, if you use a C1,C2 dual labeled probe, you have some other complications. Again, depending upon which field you're working at. This is an example of the C1,C2 labeled pyruvate in the pig heart at 3T. As you can see, now all your signals are going to be doublets. So the C1 pyruvate is a doublet, C2 is a doublet. And so your metabolites will also end up being a doublet. Not all, but for lactate and for alanine will be a doublet. The glutamate, C5, will be a singlet. But what happens is, because we are at 3T, and because of all the different doublets and the splitting of the peaks, you're going to get the C5 glutamate overlapping with your C1 of lactate. And then you would have a whole mess of peaks that's going to be really, really hard to interpret if you're using C1,C2 labeled pyruvate. But the same information you can get if you use a C1 pyruvate coupled with a C2 pyruvate. You will get the exact same information without the doublets. You will get a singlet for everything. So these are the different considerations that you need to think about depending upon which organ you're studying, what metabolic pathway you want to study, and again, the kinetics for the T1s. You may not be able to observe all the metabolites depending upon your labeling scheme. But when you're doing multiple or experiments, you have a very limited or restricted bandwidth that you can work with, especially on clinical or 3T systems. And so that's, again, something you want to consider. The most common multiple experiments we've done is the pyruvate and urea experiment. So that gives you 2 different kinds of data. One is from metabolism, and one is about the perfusion. So it'll give us a clear indicator of whether our tumor is perfused, which regions are perfused in your tumor. If you get a signal dropout, if you're doing a CSI and you don't see any signal in one specific voxel, and if you see pyruvate in that area or urea in that area, you'll be able to evaluate if that particular region of the tumor was actually perfused. But in a lot of cases, pyruvate should give you similar information in terms of perfusion. Or you could have different metabolic probes in one experiment. So in this particular model, we used a very, very aggressive patient-derived renal cancer implanted in the kidney, and it grew quite well. And we polarized pyruvate and DHA together, and we were able to get from that voxel both the vitamin C and lactate products. So again, as you can see, you need to make sure that there is no overlap of peaks in that. So if we were to do glutamine and pyruvate in the same experiment, there is absolutely no way of delineating the different probes and substrates and products. So you need to make sure that the spectral overlap is not a problem for your choice of probes. And so the C1 pyruvate has been used to verify the different tumor grade. We've shown in the tramp animals, higher the lactate production, higher the grade. It's been used by a lot of works to show response to treatment. Lactate is a very good readout of radiation effectiveness and chemotherapeutic efficacy. One thing, again, I'd like to come back to is people compare this to PET a lot. But there's actually been, other than this one work that we did with the cells, there's been no other comparison. So the question in the field is it going to give us similar answer? Is it going to give us better information than PET does? Or is it not a biologically relevant model just from the sheer volume or the concentration of probes that's required, whereas PET uses nanomoles and keeps it really lower in terms of toxicity, in terms of the endogenous concentration. And so it doesn't alter the inherent equilibrium and kinetics of the metabolism. And so, but this work here, we were able to use the exact same bioreactor, exact same cells after a hyperpolarized experiment in a PET setting. And we showed that when these PC3 cells were treated with NAMT inhibitor, which is a drug that prevents the resynthesis of NAD. So if you remember, the LDH enzyme requires the cofactor NADH to convert pyruvate to lactate. And the NAMT inhibitor prevents the NAD regeneration back to NADH. And so what happens is your lactate production should reduce with treatment. And so PET gave identical information. But what we got better was we saw a concomitant, if not more, production of alanine, but that's not something that you could ever do with PET. Get simultaneous other metabolic information, right? So the PET equivalent of our experiment would be if this was done with FDG, I'm sorry. So FDG is taken up by the cell, but it does not get any more metabolites once it's phosphorylated, and it's stuck. And so all we are getting a readout is the uptake of the probe. And so that's, again, something that you want to consider if you want to show the advantages of doing a hyperpolarized experiment over conventional or currently used modalities. It's been used for as a marker of inflammation, as shown by McKinsey's work here. So the T is the tail. This is an arthritic model, and inflammation was induced in the right hind paw of a rat, and so the left is the control. As you can see, this lactate to pyruvate ratio shows increased lactate production in the arthritic limb, which has known inflammation. And so it can also be used as a readout of your genetic status without actually interrogating the tumor, as shown by Miriam's work, where you show actually in this specific case that LDH is downregulated. So when you actually use hyperpolarized pyruvate, you do not see a buildup of lactate, which is the most common underlying theme of most cancers. But if you have this genetic mutation of this IDH enzyme, then your lactate is not going to be produced. And that right away, without having to do any invasive assessment of the tumor, you could get genetic background of the type of tumor you're dealing with. And of course, the clinical application, we have applied it to the prostate cancer patients, which we are taking forward for the phase 2 clinical studies next month. And here is just most of the probes that's been used and the common functions that's been used at. But pyruvate is hands down the single most commonly used probe to date. So that's all I have to say. If you have any questions, not necessarily I'll have all the